Introduction. The code that isn't fully cracked. In 1953, James Watson and Francis Crick unveiled the double helix structure of DNA, and the world was told a simple, comforting story. DNA is the blueprint of life, a code written in 4 letters, A, T, G, and C, that spells out the instructions for building every living thing on Earth. By the 1960s, scientists had cracked the genetic code itself, discovering that groups of 3 DNA letters, called codons, correspond to specific amino acids, the building blocks of proteins. It seemed for a while that biology had found its Rosetta Stone. Read the sequence and you would understand life. More than 70 years later, that story has grown far more complicated and far more fascinating. The genome, it turns out, is not a simple instruction manual read from start to finish. It is something closer to a vast, layered self-editing library filled with sections scientists still cannot fully translate, genes that switch on and off in patterns no one can fully predict, and enormous stretches of DNA whose purpose remains genuinely mysterious. The Human Genome Project, completed in 2003, was supposed to give us the complete parts list of a human being. Instead, it opened the door to an entirely new set of questions, many of which remain unanswered today. This article explores the strange, hidden corners of the genetic code, the parts of our biological instruction manual that continue to baffle even the world's leading geneticists, molecular biologists, and bioinformaticians. We will look at junk DNA and the ongoing argument over what it actually does, the epigenetic code that sits on top of the genetic one, the mystery of gene regulation, the challenge of predicting how proteins fold from their genetic instructions, the enigma of non-coding RNA, the unresolved riddles of genetic dark matter, and the tantalizing possibility that the code itself may be more flexible and context-dependent than we ever imagined. The basics. A quick refresher on the genetic code. Before diving into the mysteries, it helps to remember what we do understand. Human DNA is composed of roughly 3.2 billion base pairs, organized into 23 pairs of chromosomes. Only about 1% to 2% of this DNA directly codes for proteins. The molecules that do most of the work in our cells, from digesting food to fighting infections to contracting muscles. The classic genetic code works like this. DNA is transcribed into messenger RNA, mRNA, which is then translated by ribosomes into chains of amino acids. Each 3-letter codon corresponds to 1 amino acid, or to a stop signal that ends the protein chain. There are 64 possible codons, but only 20 standard amino acids, meaning the code is degenerate. Multiple codons can specify the same amino acid. This part of biology, discovered largely between 1961 and 1966, is well understood and has been confirmed across nearly all known life forms, from bacteria to blue whales, which is itself remarkable evidence of a shared evolutionary origin. But this well-understood core represents only a small fraction of the genome, and an even smaller fraction of the questions scientists are still trying to answer. The riddle of junk DNA. For decades, the roughly 98% of the human genome that does not code for proteins was dismissively labeled junk DNA. The assumption was that this vast majority of our genetic material was evolutionary debris, leftover viral insertions, duplicated sequences, and broken genes that no longer served any purpose but had never been cleared away by natural selection. That assumption has been thoroughly challenged, though not entirely resolved. In 2012, the ENCODE project, Encyclopedia of DNA Elements, a massive international collaboration, announced that as much as 80% of the human genome shows some form of biochemical activity, meaning it is transcribed into RNA, binds to regulatory proteins, or shows other signs of being biologically active rather than inert. The announcement made global headlines, with some scientists declaring that the idea of junk DNA was dead. DNA might be transcribed into RNA by accident, simply because RNA polymerase, the molecular machine that copies DNA into RNA, is not perfectly precise. Evolutionary biologists pointed out that if 80% of the genome were truly functional, the rate of harmful mutations accumulating each generation would be so high that human populations would face an impossible genetic burden, a problem sometimes called the genetic load paradox. The truth, most scientists now agree, lies somewhere in between, but exactly where remains unclear. Some non-coding regions, clearly do matter. They regulate when and how strongly nearby genes are switched on. They help organize the physical folding of chromosomes inside the cell nucleus, and they produce functional RNA molecules that do real biological work. Other regions may genuinely be inert, or may have functions so subtle, so context-dependent, or so specific to certain cell types and life stages that we simply have not detected them yet. Consider transposable elements, often called jumping genes. These are DNA sequences, originally derived from ancient viruses, that can copy and paste themselves throughout the genome. They make up nearly half of human DNA. For years, they were considered pure genetic parasites, freeloading sequences that had colonized our genome over millions of years, contributing nothing useful. Now we know that some transposable elements have been domesticated by evolution and repurposed as functional regulatory switches. Some are essential for early embryonic development, and some even contribute to the immune system's ability to fight viruses. Yet the vast majority of the roughly 4.5 million transposable element copies in the human genome have no confirmed function, and no one knows how many of them are silently doing something important versus how many are truly vestigial. The junk DNA question is not merely academic. If large portions of non-coding DNA do have hidden functions, it means our current understanding of genetic diseases is dangerously incomplete, because we are largely searching for disease-causing mutations only within the small protein-coding fraction of the genome, potentially missing critical variants hiding in the dark 98%. Gene regulation. The code. Behind the code. Even within the well-understood protein-coding genes, one of the deepest mysteries in modern biology is not what the genes say, but when, where, and how strongly they are expressed. Every cell in your body, from a neuron in your brain to a skin cell on your fingertip, contains the exact same DNA sequence. Yet these cells look and behave completely differently because different genes are switched on or off in each cell type. This is the mystery of gene regulation, and it is arguably more complex than the genetic code itself. Gene regulation works through an intricate web of molecular switches. Regions of DNA called promoters and enhancers act like dimmer switches and on-off buttons, controlling how much of a given gene's product gets made. Proteins, called transcription factors, bind to these regulatory regions, sometimes activating genes, sometimes silencing them. What makes this fiendishly complex is that enhancers do not have to sit near the gene they control. They can be located hundreds of thousands of DNA letters away, and the genome has to fold in three-dimensional space, forming loops, so the enhancer can physically touch the gene's promoter. Scientists still cannot reliably predict, just by reading a DNA sequence, exactly which enhancer controls which gene, or how strong that control will be under different conditions. The three-dimensional architecture of the genome inside the nucleus, how it folds, loops, and organizes itself into structures called topologically associating domains, is an active and rapidly evolving area of research, and much of it remains a black box. Projects using advanced techniques like Hi-C, which maps which parts of the genome physically touch each other, have revealed a stunningly intricate structure, but translating that structure into predictable rules about gene expression remains elusive. This matters enormously for medicine. Many diseases, including cancers, are not caused by mutations in the protein coding sequence of a gene at all, but by mutations in the regulatory regions that control when and how much of that gene gets expressed. A single letter change far outside a gene's coding region can dramatically increase disease risk by disrupting an enhancer, yet identifying these regulatory mutations is far harder than spotting a mutation inside a gene itself, because we do not have a complete map of which regulatory elements control which genes. Epigenetics, a code on top of the code. Perhaps the most striking discovery to complicate the simple picture of the genetic code is epigenetics, literally above genetics. Epigenetic modifications are chemical marks added to DNA or to the proteins, called histones, around which DNA is wound without changing the underlying DNA sequence itself. The most well-studied of these is DNA methylation, in which a small chemical tag called a methyl group is attached to certain DNA letters, typically silencing the nearby gene. What makes epigenetics so remarkable and so mysterious is that these marks can change during a person's lifetime in response to environment, diet, stress, and other factors, and in some cases, they appear to be passed down to offspring, a phenomenon called transgenerational epigenetic inheritance. Studies of survivors of famine, extreme trauma, and other severe stressors have suggested that the epigenetic marks shaped by these experiences might influence the health and stress responses of children, and even grandchildren, though the extent and mechanisms of this inheritance in humans remain hotly debated and far from fully proven. The epigenetic landscape of a single human cell involves multiple overlapping layers, DNA methylation patterns, at least a dozen distinct types of histone modifications, each of which can influence gene expression in different ways, and the physical packaging of DNA into tightly wound structures called chromatin, which itself controls whether genes are accessible to be read at all. Scientists have begun to speak of a histone code, a set of combinatorial rules by which different chemical marks on histone proteins interact to produce specific patterns of gene expression. But unlike the elegant simplicity of the DNA to protein genetic code, the histone code appears to be combinatorial and context-dependent in ways that resist simple rules. The same modification can mean different things, depending on which other modifications are present nearby, which cell type it occurs in, and which stage of development the organism is in. This is one of the frontiers where scientists still don't understand, is the most honest and accurate description of the state of knowledge. We can catalog these epigenetic marks with remarkable precision using modern sequencing technology, but predicting their combined functional effect. And understanding exactly how environmental signals get translated into specific epigenetic changes in the first place remains one of biology's great open problems. The mystery of non-coding RNA. For much of the 20th century, RNA was viewed mainly as a passive messenger, a temporary copy of DNA's instructions, ferried to the ribosome so a protein could be built. That view has been overturned. We now know the human genome produces a staggering variety of RNA molecules that never get translated into protein at all, yet perform crucial biological functions of their own. These include microRNAs, tiny RNA snippets that can bind to messenger RNAs and block them from being translated, effectively acting as a fine-tuning volume control on gene expression. There are long non-coding RNAs, LNC RNAs, some of which are thousands of DNA letters long, that can act as scaffolds, organizing proteins into functional complexes, or that can guide epigenetic machinery to specific locations in the genome. There are circular RNAs, whose closed-loop structure makes them unusually stable, and whose functions are still being actively investigated. There are Peewee interacting RNAs, small nucleolar RNAs, and a growing list of newly discovered RNA categories, many named more for their initial physical characteristics than for any thorough understanding of what they do. The scale of this hidden RNA world is enormous. Estimates suggest that tens of thousands of long non-coding RNAs are produced by the human genome, yet the specific function of the vast majority remains uncharacterized. A landmark study once estimated that pervasive transcription, meaning RNA is copied from far more of the genome than was previously believed possible, is the rule rather than the exception. Whether all of this transcriptional activity is meaningful, or whether much of it is transcriptional noise with no biological consequence, is a question that remains unresolved and is likely to differ RNA by RNA. What is clear is that this hidden layer of RNA regulation adds a staggering amount of complexity to the genetic code. If DNA is a script, RNA is not merely an actor reading lines. It is also a director, a stage manager, and sometimes a character in its own right, and we are only beginning to map out its many roles. The protein folding problem, partially solved, still mysterious. Even when we know exactly which amino acids a gene's protein coding sequence will produce, a profound mystery has historically remained. How does that one-dimensional chain of amino acids fold within milliseconds into a precise three-dimensional shape that determines its function? This is known as the protein folding problem, and for decades, it was considered one of the most significant unsolved problems in all of biology. In 2020 and 2021, this picture changed dramatically with the emergence of AlphaFold, an artificial intelligence system developed by DeepMind, which was able to predict protein structures from amino acid sequences with an accuracy rivaling experimental methods for a large proportion of proteins. This was a genuine scientific breakthrough, and it has already accelerated research into diseases, drug design, and enzyme engineering. But important mysteries remain even here. AlphaFold and similar tools predict a protein's most likely final folded shape, but many proteins do not have a single fixed shape at all. They are intrinsically disordered, shifting between multiple conformations depending on their environment, or folding differently depending on what other molecules they interact with. Understanding these dynamic, shape-shifting proteins remains extremely difficult. Additionally, predicting a protein's static structure is not the same as understanding how it folds, the actual step-by-step physical process, the pathway a protein takes as it collapses from a floppy chain into its functional form, remains only partially understood, and predicting how proteins interact with one another in the crowded, complex environment of a living cell remains a significant ongoing challenge. Genetic dark matter. Unknown genes and unassigned functions, biologists and bioinformaticians. We will look at junk DNA and the ongoing argument over what it actually does, the epigenetic code that sits on top of the genetic one, the mystery of gene regulation, the challenge of predicting how proteins fold from their genetic instructions, the enigma of non-coding RNA, the unresolved riddles of genetic dark matter, and the tantalizing possibility that the code itself may be more flexible and context-dependent than we ever imagined. The basics. A quick refresher on the genetic code. Before diving into the mysteries, it helps to remember what we do understand. Human DNA is composed of roughly 3.2 billion base pairs, organized into 23 pairs of chromosomes. Only about 1 to 2% of this DNA directly codes for proteins. OS, the molecules that do most of the work in our cells, from digesting food to fighting infections to contracting muscles. The classic genetic code works like this. DNA is transcribed into messenger RNA, mRNA, which is then translated by ribosomes into chains of amino acids. Each three-letter codon corresponds to one amino acid, or to a stop signal that ends the protein chain. There are 64 possible codons, but only 20 standard amino acids, meaning the code is degenerate. Multiple codons can specify the same amino acid. This part of biology, discovered largely between 1961 and 1966, is well understood and has been confirmed across nearly all known life forms, from bacteria to blue whales, which is itself remarkable evidence of a shared evolutionary origin. But this well-understood core represents only a small fraction of the genome, and an even smaller fraction of the questions scientists are still trying to answer. The riddle of junk DNA. For decades, the roughly 98% of the human genome that does not code for proteins was dismissively labeled junk DNA. The assumption was that this vast majority of our genetic material was evolutionary debris, leftover viral insertions, duplicated sequences, and broken genes that no longer served any purpose but had never been cleared away by natural selection. That assumption has been thoroughly challenged, though not entirely resolved. In 2012, the ENCODE project, Encyclopedia of DNA Elements, a massive international collaboration, announced that as much as 80% of the human genome shows some form of biochemical activity, meaning it is transcribed into RNA, binds to regulatory proteins, or shows other signs of being biologically active rather than inert. The announcement made global headlines, with some scientists declaring that the idea of junk DNA was dead.